with Clafter, and he works with also with many other people, but he's also interested in life and biology application of single particle trajectories. Ralf, please. We're happy to welcome you here.
Thank you all. Thank you, David, for the introduction. I missed this one, and I feel so sorry. But you see, I corrected the date. And I changed a few slides of the talk, but as you can see, this is obviously not Manchester. This is annoying that I hear myself. This is a beautiful city with the sunshine of Cambridge. It's gleaming, isn't it? So we organize a beautiful meeting there again this October, and I have to travel there all the time to prepare the meeting. What I want to tell you about is anomalous diffusion, and in a very simple sense. So I want to define it in the sense of a mean square displacement of a stochastic process, and the mean square displacement in time goes like this power law. So instead of alpha equals 1, the typical Brownian motion, I have some alpha here. I will mainly concentrate on cases when this alpha is smaller than 1, so the diffusion is less efficient than Brownian motion. I will also show you some examples for super diffusion when alpha is larger than 1. And typically when we talk about stochastic processes, this is the quantity we're having in mind. That's also when physicists want to model complex systems, this is a quantity that's easy to calculate. And in this sense here, the ensemble average is just a spatial average over the second moment of the spatial coordinate multiplied by the probability density function. However, when you have single particle trajectories from simulations, from experiments, and typically you have few of them, but long trajectories, they range in my notation from 0 to some observation time, some measurement time. And then typically in these papers people calculate the time averaged mean square displacement. And you can see what you do, it's kind of a sliding average. You introduce a lag time, and then you look at the difference of the particle position separated by this lag time, you square it, and then you integrate over this quantity over your entire time series. So here in the ensemble average, the mean square displacement is a time local quantity, whereas here you take the information of the entire trajectory into account. I said you integrate, you normalize by this guy, and when we talk about anomalous diffusion processes, in what follows, I want to concentrate on two of these processes. I mean, what's clear, this guy, this is no longer under the spell of the central limit theorem, so there may be many physical, or if you want, mathematical processes that give rise to some kind of a t to the alpha, and I only want to introduce two here. One is called fractional Brownian motion, so that's for Western literature going back to Mandelbrot and Van Ness in '68. If you have Russian friends, they tell you, oh, there was Russian literature. Kolmogorov, 55, already looked at this. Fine. Physically, of course, what I should write down is a Langevin equation that actually has a mass that has a friction where you couple the correlations of the noise with the friction, and you get memory terms. But in the overdamped limit that I'm interested in here, this fractional Brownian motion is sufficient to describe the process. It's in that long time range, it's equivalent. So just look at the Langevin equation where we have the derivative of the position, we have the driving noise. This noise is supposed to be Gaussian. However, you see it's long range correlated. So when I have these two times here, we have this alpha to the minus, we have the exponent alpha minus 2. And especially when we have a subdiffusive process that I want to put here, we have a minus sign. And you will see that afterwards, this will cause a very sharp dip in the increment correlation function. It means that when you move in one direction, the next step, if you want, is going in the opposite direction. So you have this very strong what Mandelbrot called anti-persistence. Very importantly, when you start with this Langevin equation, you calculate the correlation functions. You can show that in the limit of sufficiently long observation times, this quantity converges exactly to this T to the alpha we have from the ensemble average. And for us in physics, we call this ergodic. Now, for the mathematicians of you, this is not what you understand by ergodicity. So I'm not talking about mixing behavior. I'm really only referring to this Boltzmann sense of ergodicity, equivalence of a time and an ensemble average. And we will see that there are other processes where this is not fulfilled anymore. And actually, there's quite a number of complex systems where this property that physicists get brainwashed with in their studies is actually violated. And we will see two consequences. So one is, of course, that when this is violated, we cannot describe a time average in terms of the ensemble average, but we will also say that time averages may become random quantities, not because of a complex environment, but because of the dynamics that's encoded in the system. So this other one, this other process that's very suspicious, as you can see, is called continuous time random walk, and that's essentially going back to Montreal and Harvey Shear in a paper in the early 70s. And they assume just a regular random walk process. Of course, they take a continuum limit afterwards, but it's a random walk process in which consecutive steps are separated by a so-called waiting time. So in a primitive sense, you can imagine a random energy landscape. Your particle is moving, your random walk is moving, falling into an energetic trap, has to escape, falls into the next trap. And if these traps, one can show, are exponentially distributed, meaning you can always find a deeper trap to fall in, this translates, just by the Kramer's rate, into this power law of waiting times. And you see when alpha is in between 0 and 1, there is no characteristic waiting time anymore. So we have a scale-free distribution of waiting times, that will show you experimental examples that these really exist. And in this case, you can show that now the time and ensemble averages, they will not converge, even if you take your measurement time to infinity. And the reason is exactly this scale-free nature. When you draw more and more waiting times for every step, you can get longer and longer waiting times. So a single or a few extremely long events, they can dominate the whole time series. Okay, and as I said, this quantity actually remains random. It's a non-stationary process, and we will see it also depends crucially on its age.
So by non-stationary, do you mean that the initial conditions are never forgotten, or something else?
In this case, yes. You could also think of non-stationarity when, for instance, you have a diffusion coefficient that becomes a function of time. Right? You have a
Sure. I'm just asking what sense is what causes it? You know?
Here, it's a very strong memory of the initial condition. Absolutely, because you have this very strong probability that the particle never moves. And this was the big puzzle when these people started to understand how photocopiers work. You have these semiconductors in there, and none of the experiments could be understood with the old theories. Anyway, and what I'm going to tell you about, we have in this physics today, and I actually sat down 2 years back and wrote a long review article. Let me start with a relatively simple system. It's very colorful, so this is nice for starting a talk. So these are caricatures for biological membranes. In this case, they're bilayers made up of one single chemistry of lipids. So you have lipid molecules, they have a head group. The head group is hydrophilic, likes the water. And out of this head group, you have two tails, and these are hydrophobic. And then what happens, so the water would be this, I don't know, this stuff at the top and bottom. So they would arrange in this way as a double, as a bilayer to avoid contact of the tails with the water. And then you see this is quite disordered. You have a lot of entropy in there. This is a snapshot of the simulations, and the different colors, they just mark different lipid molecules, but remember they're all of the same chemistry. So this is probably what happens when you're at a party last night. People were playing poker at our place, they were sitting, but if they had been dancing, that would be the analog for that. You can do two things to the system, and the first thing is you can actually include these white molecules here. These are cholesterols. This is the stuff that doctors want to make us believe is bad. but actually our membranes wouldn't work without these cholesterol, so we need to eat the good stuff. But you can see there's a lot less entropy in those. So they occupy available space in between these tails. They really fit in nicely into these tail groups, and they take out a lot of entropy, so the system becomes less mobile, and you see as a price also the width of your membrane is increasing. So this would roughly be corresponding to the Danish royal guards when you go to the Danish palace and you see the beautifully dressed soldiers there guarding the queen, but you know, oh, there's my nephew, okay, you wave, right? These are Danish soldiers. You can do another thing. You start with this membrane, the happy membrane, and you quench temperature. And then you go through a phase transition and you get to a gel phase. And I was just in London 2 weeks back, and this is how the good London British soldiers, right? They are very strict and stiff. And you can see you don't need anything, you just have the lipid chains, and they're almost immobile and form this gel phase. Now, this phase was studied previously very extensively, and I want to mention that one first. But then our question was, and this is a collaboration with the group of Ilkka Vattulainen in Tampere, who's a big simulation guy. Our question was, is the motion in these two phases qualitatively very different? Good. So let's first look at typical trajectories. This is in this happy phase, the party phase, where you have different lipid chains and they're just fuzzing around. You can see they can swap positions. So it's really a liquid-like structure. And in all of these images, the edge of one of these squares is 1 nanometer. When you go to the Danish phase, when you have the cholesterols, you see that the motion is within the same time frame, time window. The motion shows much less mobility, but you also see that qualitatively the motion of cholesterols and lipids is the same.
So, just 1 second. Can you describe? So, this is a simulation of one of these lipids that you see
in the simulations. Yeah. Actually, you have the trajectories of all of them.
Okay.
These are just a few sample trajectories.
Okay, and what's the interaction? Do they interact, or this is just exclusion? How do they move?
Well, it's more or less excluded volume.
So this is the only force?
There's no force in there.
No force?
It's just a stochastic motion.
And this is 2D, or this is also account for the 3D geometry?
So what you have here, of course, you look at the projection of these guys, but it's a full 3-dimensional simulation. I should have said that. It's molecular dynamics. It's all atom. And don't ask me about the number of water molecules we have here, but there were sufficient water molecules.
And the water molecule can interact?
It doesn't penetrate into the membrane. So
But
it's really being expelled. They interact, but it's only at the surface. Because there's a very strong repulsion of the water by the tails. So we have these trajectories, and you see in the gel phase, there's really hardly any motion. Now let's look at the mean square displacement. So in this case, it doesn't make a difference because it's an ergodic system. So I could as well plot here the ensemble average. But what you can see here, this was known all the time for this system, and we tried 3 different chemistries of the lipids. Each time, all the lipids have the same chemistry, but we tried 3 different chemistries. Essentially, the difference is, from a physical point of view, when you look at these lipids from above, their cross-section is a little bit different. Due to double bonds, chemistry, I can't even pronounce these complicated names. Doesn't matter, but you see there's anomalous diffusion here, an exponent around 0.6, but until a ridiculously short time of maybe 10 nanoseconds, and this is roughly the time scale over which these guys, nearest neighbors, exchange positions with each other. And this is enough to restore more or less normal diffusion. We have 0.97 to 0.94, essentially. Once you put the cholesterol in there, you can see that initially the exponent is a little bit lower. Here it's only 0.5. And then there's a crossover only for one lipid chemistry. The other 2 have an exponent around 0.8. So already here you can see you add a little bit of disorder in terms of these cholesterols, and you extend the anomalous diffusion regime. And when you go to the gel phase, it gets very extreme. they almost stall at short times. You see it's below 0.2, the exponent, then it crosses over to something like 0.6.
So you're never resolving a ballistic regime here then?
Not here, no. When you focus in and you make the time step smaller of the integration, you can see the ballistic part.
And what's the parameter that they are changing in the simulation to generate disorder, order versus?
Well, here you stuff additional molecules in between the tails. So here this is essentially taking away degrees of freedom and also making it more difficult for them to move past each other. And here you just change temperature. Well, to be honest, simulation guys, they also need to change the force field because otherwise it doesn't work. There are many subtleties, but essentially this is a phase transition that you can map experimentally. So it's a well-defined phase transition, only depending on the chemistry of the metal.
And you can't measure the MSDs experimentally, I guess. Not at that. Can you measure the MSDs at that time resolution?
Well, we have essentially the trajectory. We don't measure, we do the simulations, right? So we have a recording of all the positions.
But at what time resolution? Experimentally, I mean, not.
Oh, experimentally, yes, this is different. I will show you experiments later on, but there you're more on the millisecond range. So at the other place in Oxford, there's the group of Christian Eggerling. And they can actually monitor the motion of individual lipids in membranes, but of course on longer time scales.
So what's diffusion coefficient doing here? If MSD is going slowly and speeding up, diffusion coefficient slow and faster?
It depends what you mean by diffusion coefficient, right? So here in the normal range, of course, it's clear it's a normal diffusion. And you can see that it's also going down. I have it in the paper, I forgot the numbers. And of course, you can also define a proportionality coefficient for the anomalous case, and this is also affected by the temperature decrease of the cholesterols. Yes. So it seems consistent that it's you don't only get slower in terms of exponent, but also in terms of prefactor.
But at short times, though, is it actually diffusion coefficient faster or slower than at long times?
How do you compare really, right? The one has units of centimeter square over second, the other one centimeter square over second to the alpha.
Right.
So you can say, okay, I want to compare them at exactly 1 second.
Right.
And I don't have the answer ready.
So I'm curious how to interpret that plot there. They're right. Sort of, what's it mean to have a low MSD power at first and then higher MSD power? What does that mean?
Oh, it just crosses over. We don't exactly understand what happens physically. So far, this is just an observation. And our question, we don't have a good model that describes the crossover. So in this case, we're just trying to essentially use a fractional Brownian motion model, but we temper the power law. So we have an exponential cutoff of the power law. In this case, you might think that you have, like in polymer physics, you could have two power laws, one crossing over to the other. It's possible. The real physical mechanism we don't understand. I mean, this is why we do simulations for these quite complicated systems. That's where the field is.
Good. Just a question. What is actually the difference in the simulation for the white molecular? I mean, the one case you have you assume you have you use time steps and then go from one time to another, which is maybe 10 to minus 1 nanosecond. I don't know how much. So what's the difference from liquid disorder time step, because you have in the one you have liquid order, you have some cholesterol additional, so you also, but the thing is just you choose some Gaussian number like this and then you move it, or and then you see if it's free.
Essentially, you
or not.
couple it to a thermostat. Yes. So you have a well-defined temperature of the system.
Yeah.
And then it's using all these software packages like Romax, and then you have to plug in the force fields, and that's the delicate part.
So you already put something with exponent alpha, this kind of correlation function. Is it something input, or this is just something?
No. This is just an observation. This is just an effective analysis of what we see.
Yeah.
So you plug in first principles. These are regular equations of motion plus a thermostat.
Yeah, we have to put some force, no?
Yes, I mean you have interaction forces. You have Hamiltonian. It's a well-defined system. And then we run the simulation, and then we analyze the trajectories. Because that's the only thing we understand. The Hamiltonian is horrible, the force fields are horrible. You have, I mean, on this resolution, I mean, you have really all atoms. So these blobs here, they have an internal resolution in turn. It's not an easy system, but the funny thing, and I don't show you all the plots, but what we see, first thing, because we came from this non-ergodic motion, the first thing we were interested in was the ergodicity. So here you see 128 trajectories superimposed on each other. This is roughly the, I think this is the observation time. So of course, when your lag time approaches this guy, you get all the scatter, so this is clear. But you see for all the earlier lag times, we have a perfect superposition of the trajectories. So they're ergodic, they're reproducible. The anomalous diffusion that we see here is perfectly consistent with the ensemble average. Now the next one, we look at the correlation function, the two-time correlation function of the increments of the motion. That's what I said before. You start at 1 and you have this drop through 0, you go to a negative value, and then when you use the model, this fractional Brownian motion, you have a power law recovery back to 0. So this is a very characteristic feature of this anomalous diffusion. This is for a larger alpha, and this is for the gel phase here, where you see that the shape changes a little. This would be for something around 0.15. But you see that in all the cases here for the lipids and the cholesterols, here for the lipids, the agreement with the theoretical black line for the theoretical fractional Brownian motion is very nice. At that time when we started, there was still kind of another model for the motion of the lipids. The idea was that you have a void. Don't ask me about the color. Let's say these red guys here, they would represent the lipids, and the yellow one would represent a void. So this void would then, change places with the lipids, and this would create the lipid motion. So, however, you can imagine this guy only moves when this diffuses here, so you have a power law waiting time for making a step. So this would actually be one of these continuous time random walk models. And another way, so this is already pointing against this, another way of how we analyzed it is that we looked at the first passage of a lipid that starts in the middle of a circle and until it crosses the border of the circle. And for continuous time random walks, you would expect power law decay of this first passage time density, but what we observe is perfectly exponential, consistent with the fractional Brownian motion. Right. So this was the first case where we only have the lipids. Of course, realistic systems are much more complicated, and the first step towards these more complex systems is that we put in big chunky proteins. So these are proteins, they feel happy among these lipids. You have realistic membranes. I mean, all our membranes, they're filled with all kinds of protein channels, whatever, they're really crowded. I will show some pictures afterwards. But in order to simulate the system with a large enough number of these guys, we actually now have to move to a coarse grain simulation. So now it's these Martini force fields, if you know. And then we look at what happens when we increase the concentration per lipid of these proteins. So for the long time motion that we now can access because of this coarse graining, we look at the diffusion coefficients of the lipids here and of the proteins over here as a function of the concentration of proteins. So this is the completely dilute case. Here you would have a measure for the diffusivity, and then we plug in the proteins, and you can see that the lipid diffusion is strongly affected. It's going down significantly. And I should also say that we have two kinds of lipids here, and I forgot which one does what. One of them causes the proteins to aggregate, so then they cluster, and the other one allows them to more or less move free. And I think the red one should be the clustering, because when I look at the diffusivity of the proteins, it more or less goes down to 0 at a high enough concentration for the clustering one, but still drastically for the others. one of the measures we looked at, it's not a very good measure, but it gives you an impression of what happens there. We looked at the local anomalous diffusion exponent, so that's just from the logarithmic derivative of the mean square displacement. And again, from top to bottom, you increase the protein concentration, and you can see that there's also very strange crossovers in the motion. So it almost starts to speed up, and then it actually goes down again, has this very characteristic dip. Again, no physical real understanding here. And then the postdoc who did the simulations, he just said, okay, if I'm a little bit careful, I extrapolate, or the system will meet normal diffusion again. And you can see now we're already in the range of microseconds. So we already came from nanoseconds of the pure lipid system to something like microseconds. So the more disorder we add in that system, we increase the range of the anomalous diffusion.
So how is the dash line? How did you do the dash line?
This was a good guess of the postdoc. There's no
simulation,
no systematic simulation or anything behind. He just said, okay, let me continue with a slope here, and then I require that I don't ask me for details, I don't know. You see, I was not the senior author. Anyway, up to here, where you have the full lines, these are the data. Okay. And then, very recently, they managed to increase the simulation time by another factor of 20. I'm always amazed how much money you need to run these simulations if you have to pay for the computing time. So again, you see it's the same system, just more fancy pictures this time. Here we have a case where we have all those lipids, and we have one protein sitting in there, and then we have a protein case where the proteins do not aggregate, and here you can see that they form aggregates. And just to give you an impression, the all-atom case would be the backbone of this lipid chain, and in the coarse-grained model, we only have these blobs. And these would be the proteins. Very faintly, maybe you can see the full structure of the protein behind, and then this glossy structure on top. This would be the shape of it in the coarse-grained simulations. So again, the anomalous diffusion we can all say, but the striking fact this time was that we had enough data to also reconstruct the probability density function of these guys. And I told you in the dilute membrane it's perfectly Gaussian. This is what you also see here. This is 1 minus the cumulative distribution. And if we plot it in this log-log scale, you can really see the we take the logarithm of it. So it's really this square scaling that represents the Gaussian behavior. Once we go into these crowded membranes, we actually realize that the Gaussian nature is lost, and we could more quite nicely actually fit the data with this, if you want, stretched Gaussian law, where the stretching exponent is clearly different from 2. Don't ask me what causes this exactly. We have one clue that I show you on the next slide, but it's clearly showing you that this assumption of the Gaussianity failed in this case. And it's also failing for the protein case. There you also get deltas down to 1.0. Let's look at the motion of the individual trajectories. Here we see the lipids, so they all fall together, and these are the proteins. They're just slower, of course, they're bigger, they have a lower mobility. But see what happens when we go from the dilute case to the one with many proteins. Now we can see that many of the lipids, they're very closely associated to the proteins. They get stuck for some time. So we get a much more heterogeneous type of motion. And it's still only one chemistry of lipids. Imagine we have many different lipid chemistries on different proteins in there. It would be a total mass. And the bright line here, this is just the ensemble average over all the lipid trajectories. And here you also see the scatter of the motion of the proteins. And in the clustering case, you can see that also the mobility of the lipids is going down a lot. And this is, of course, due to many that even get caged and so on. Let me skip this one quickly. Funny enough, when you take an extremely simple model, and this is of a model of small spheres, if you want. So we use argon in these simulations. So we have essentially a dense system of these argon molecules, and if we look at the motion, it's beautifully Brownian, whatever, Gaussian diffusion. But now we add obstacles. So we take immobile argon particles and form these red obstacles. My students never tell me the colors of the images. Anyway, so when we have this kind of obstacles, things still move nicely. As soon as we create more closed spaces, what you can see is, or maybe not, it's very faint here, that for the dilute case, the probability density is very nice and Gaussian. Once you add these obstacles, you get these deviations from the Gaussian, and you actually need stretch Gaussians to describe the motion. I don't claim that this is all the effect that we see in the real system, but already in this very simplistic system, when we create fractions of more mobile and less mobile particles, we already get deviations from Gaussian behavior. That's all I say. Good. Now we come to real. Oh, do you have a question here?
Is it,
Yes.
we usually see kind of, I mean, it's kind of even sharper, kind of.
This wiggle here,
Yes, so it's even more around zero than usually.
I suspect this could be some that are just squeezed in here.
Yeah. Kind of, can you go back to what you saw? So,
Yes.
so your other
Wait.
Gaussians, non-Gaussians were, what shape
Yes.
were they?
I don't have
You
the
haven't
real PDF here. It's only this integrated one. We can check the paper afterwards. I have some with me. Yes. I don't recall whether there's a wiggle or not.
But is
We
it, do
can
you ever
check.
have it that it's flatter than a Gaussian? Or probability distribution?
Wait, when you make the tails higher, you should also flatten down the peak.
Yes.
Yes,
Is that what you see?
that would be my guess.
Okay.
At least how I can preserve probability. Good. Now let's go to the real membranes. So now we add all the complexity, all the shit of a living cell. And the first time I saw this paper, I was thrilled because we had worked on all these strange anomalous processes. We calculated quantities, and then came a paper, and they could show all this. So I was really taken by this. So this is from the group of Diego Prof at Colorado State University, and they managed to follow trajectories of fluorescently labeled potassium channels in the membranes of living cells. So membranes, this is nice to trace because you have a more or less flat surface, right? So they can actually follow the particle for very long trajectories. This is 1 micron, so you have a span of, I don't know, almost 10 microns here, and they can record this time series for hundreds of seconds. So that's quite amazing. And the first thing what they see then when they plot the mean square displacement of different trajectories, of course the window they show is not that great, but you will see that at least it's consistent. What you see is that there's a very, very wide spread of amplitudes. I told you that these are trajectories. They are measured for several hundreds, no, not here, but I see several hundreds of seconds. Here we have data from around 100 seconds, so I think they go to 300 or 400 seconds overall. So you have quite a bit of averaging when you take your time average mean square displacement. Still, you see this very large spread of 1.5 orders of magnitude. That's remarkable. The second thing what they see, and this is what I meant by stationarity, when you evaluate your time average mean square displacement at one given lag time, but you vary the length of the data you use for taking the average, you see that actually you get a scaling behavior. So the longer you monitor the system, the less mobile it appears. And this was done here for 4 different lag times. So this quantity here, this would be the lag time, and this would be, I lost the label, this would be the measurement time, and I'm pretty sure that these are 100 seconds.
The basic idea is that the longer you run, the more likely you are to sort of fall into a trap that kind of gets you
Exactly.
stuck for a time
Exactly.
comparable. Yeah.
And that's also what they see when you go to the the PNAS webpage and you look at the supplement, they have videos in here. I use full text, so I don't show videos. But what you can see is that these particles, they move happily about, and then all of a sudden they are more or less immobilized, then they just hover around a bit, and then they become mobile again. So what they do is, for these immobilized phases, they more or less measure the times. Let's call them waiting times. And when they plot the statistic of these waiting times, you see that it's relatively nicely, I mean, it's an experiment, an inverse power law. So we get exactly these waiting time distributions of the continuous time random. And what happens biologically, I don't remember the details, but these channels here, they're involved in some bigger machinery that triggers this intake of particles from the outside when these bubbles form in the membrane, and then bud off and transport the stuff inside. Endocytosis, I think it's called. So each time the potassium channel finds one of these big centers where this process is taking place, it's been immobilized. And based on these experiments, they have some papers where they talk about the biology. So what we have is, in this case, we have these waiting time distributions. We know, of course, from our theory that this gives anomalous diffusion in the sense I defined. We can also show that the time average mean square displacement looks like this. So when we calculate the whole thing, we can use the fact that this is actually a renewal process. We can go through all the derivation. It's linear in the lag time. So if you're a simple experimentalist or simulations guy, you see linear and delta normal diffusion done. Only, and this is what very few experimentalists actually do, when you vary the length of your recording time, you see that it influences the measurement. And that's exactly what you see in this plot here. So this is highlighting the non-stationarity of the process, or if you want to go back to what I said before with the traps, this is when you let the system run, you will find an ever deeper trap. So effectively, the mobility is a decaying function of time.
Can you remind me whether that linear relationship between delta squared and the other delta, is that a generic feature of CTRWs, or is that for special?
This one here?
Yeah.
You actually find it in a vast range of anomalous diffusion models. It's very characteristic. And as soon as you're non-stationary, we created, beautiful models. We even have models in which this, no, I don't have it, the ensemble average is logarithmic, like C9 diffusion or strongly disordered systems, and this guy is still linear in delta. There's only a few cases when I know that there's a delta square or a square root of delta.
What if you make a cutoff for the traps? I mean, infinite traps are somehow, and
Of course, eventually you cross over to normal diffusion. Clear. What you then say.
When this happens, at what trap energy or something you have to put in order to get this?
I'm not talking in terms of energies here. One would have to translate. So if you look here, at least over the range of the experiment, it still looks like a good power. So the cutoff, if you want, would be beyond this time scale. And it's actually not what they observe. So it's really okay to treat the system as if it had a diverging time series. We have another experimental case that we looked at where the particles are much, much bigger and it's inside a living cell. And there we had to introduce a cutoff. And what you see is that you take out all the extreme events. So the aging becomes almost a constant function. This here is it still scatters, you still have the same behavior for the mean square displacement, because that's composed of many individual steps usually. But extreme events like this aging, this is different.
So I guess the intuition is that if this delta is sort of not too big, then if you're looking, your time averaging it. No, never mind, sorry.
If you bear with me afterwards, we can check. I once had a scaling argument why this is, but I never remembered you.
Okay.
These are these arguments, you can derive everything if you're not careful on the blackboard, and I don't want to get into that.
Are all particles here mobile at time zero? Are particles here selected for mobility at time zero, or they say take all particles at time zero? Or they say just take the mobile ones and watch those?
This is actually a very good point, and let me defer it a little bit to a later point. But of course, exactly when a system is non-stationary and aging, the big question is, when do you initiate the system? Absolutely. I come back to that in a second. Let me just show you that this is not only the case in this experiment. There's some other work from a group in Barcelona. It's around Levenstein. And they look at different mutations of one given membrane protein. And then one of the key signatures they look at is, again, this aging dependence. Here they call it effective diffusivity. It would just be a measure for the mean square displacement. And you see, again, it becomes a scaling function of the measurement time. But for this one here, and this is this guy, where they cut off more or less most of the protein, and you can see that in this case it's a perfectly stationary process. So people are using this to analyze data. Let me quickly come back to this. So this is the case we just had for the free motion. What we can calculate from a renewal approach is when we look at the distribution of the quantity psi, this is the value at one given lag time of the mean square displacement divided by the average over all the mean square displacements. So if this quantity is close to 1, you would have a reproducible process. What we find for this continuous time random walk, we can calculate it, becomes one of these asymmetric Levy stable, but for instance for alpha equals 1/2, it's a half Gaussian. Levy stable doesn't matter here because the variable is in the denominator. So you still have converging moments. You can re-express it in terms of one of these Whittaker functions. Then it looks a bit less evil, if you want. So here you get this half Gaussian. This would be the case for alpha 1/2. And you see you're very far away. Most of your behavior is very far away from the ergodic behavior, so you have many cases where the trace is almost completely immobile during the measurement, and you have other cases where you have a higher mobility. When alpha, was it from outside? When alpha goes back to 1, you slowly see a peak emerging around the 1. Here you see a decay, and then eventually at alpha equals 1, you would get back to the delta function perfectly reproducible. And these are actually, in other analyses I will not show here, it turns out that this is a very, very good quantity to actually look at processes. Is it this kind of processes? Another one. So it's a very nice method to infer processes behind it. Let me very quickly highlight the fact what I said before, that we average over the entire information of a trajectory. Imagine some people, when they do single particle tracking, they don't just use a microscope, they use optical tweezers. Optical tweezers are very highly focused laser beams, and you can trap with them, for instance, a dielectric, like a micron-sized plastic beam. The reason that people use this until a few years back was that they can use these setups of optical tweezers to look at the light that is refracted back, and it goes onto one of these quadrant photodiodes that allow a much higher time and space resolution. So this was the motivation to look at this, but what you know is that on this dielectric, this trap exerts a harmonic confining potential. So it's like connecting your Brownian particle, or actually the anomalous particle, to a spring. And in this case, I said then, when you calculate the time average mean square displacement, you average over the part while it was still free, and you average over the part when it's really feeling the confinement. Imagine you start at the well of the potential. Initially, it doesn't feel a force, then it ventures out and it feels the confinement. So when you do this, at very short times, you still have this linearity of the mean square displacement, the time average, but then you cross over, once the particle feels a confinement, to another power law that's now given by 1 minus alpha. So while the ensemble average shows the plateau, that of course you would expect particle in confinement, you get to a thermal plateau, but this guy here takes both the free and the confined motion into account when you calculate the time average, and you have a continuation. Still a growth of the motion.
What's lambda 1? If you have an asymptotic inequality involving lambda 1?
This guy here, this is just the lowest non-zero eigenvalue of when you have a confining potential. So you just use separation of variables. You can do in this case And actually, I was arguing with the harmonic potential, but it's true for anything, even when you have hardware confinement. What you see here, these are actual experimental data of particles that are dielectric. They're a few hundred nanometers in size, and they're measured in a living yeast cell in the Anomalous group in Copenhagen. What you see is one type of cell, another type of cell. We just shifted on this logarithmic axis so you can see the different motions. So the amplitude here is arbitrary, but you can see for one cell you have the scatter, you have the unbounded motion, the unconfined motion, which in this time average is linear in delta, and then you cross over to this second power law, and when we look at all kinds of other quantities, it's really the kind of process I was just talking about. And this was funny, we have done the calculations. I came to visit friends in Copenhagen, and I remember Christine, we were sitting on the beautiful lakes in a coffee shop, and she had this very shyly produced these experimental results that showed this stuff, and I was fascinated to see that what we calculate can actually be measured. Good.
So, just one
Yes.
question. So now, if you have those data, now you extract alpha.
Yes.
What can you say about the biology, about the physics underlying this alpha?
I'll come back to that again. So what you see for this system, of course, I neglect all kinds of details. So this is a system where for the first time, because of these tweezer setup, they had access to much shorter time ranges than all the other volume experiments in those days. And what you can see when you look at the longer time data, they're perfectly consistent with the others. There you see more of this fractional Brownian motion behavior, and that would be a picture of the viscoelastic nature of the environment. So you can interpret the environment in which this guy is diffusing like a viscoelastic continuum. What exactly happens at these shorter times, we don't know. So it's not connected to effects of the cytoskeleton, because in the yeast cells, the cytoskeleton is very highly concentrated on close to the cell wall, and these guys were further inside the cell. So I don't think it's connected to the cytoskeleton. Other groups also showed that when you disrupt cytoskeleton, it's not really connected to it. Exactly what happens here, the physical nature of it, I have no clue. I would love to understand more about this crap. Yes. Good. But let me finish by coming back to this non-stationarity and look at the aging. So let's assume we can control the measurement. I mean, biological cells are always difficult to control. What is the start of a measurement in a biological cell? But let's assume we have another process. For instance, people show that you have the same kind of statistic when you look at blinking quantum dots or other processes. Assume we release a particle at time t equals 0, and then we have these blue impulses that would be the particle makes a step. So we let it diffuse, or we let these quantum dots blink, they go from on to off, and then we let it age. So this is a process that would follow a continuous time random walk, or renewal process with a scale-free waiting time distribution. And only here we start measuring. So when you let the system age for a sufficiently long time, what happens, you get to these longer and longer waiting time events. So it gets more and more probable that when you start looking at the particle, it's in one of these long trapping events. But you don't measure the particle exactly at the moment when it becomes immobile, but somewhere in between. And this actually changes the correlation, the probability density to measure the first step in your measurement. And by the internal memory of the system, this actually propagates along. So it changes the response of your system, if you will. The measurement will depend on when you start. This you can see when you look at the continuous time random walk, you look at the mean square displacement, the ensemble average, when the aging time is very long compared to the time you measure t, in this case would be measured from you. So this is when we measure t. You see that the alpha actually comes with the aging time, and the system again looks as if it were diffusing normally. This was the initial counter-argument against continuous time random walks for the semiconductors. In the other extreme, when your measurement time is much longer than the aging time, all this memory is already lost and it looks like normal anomalous diffusion. The beautiful thing is when you go to the time average quantity, the time average mean square displacement, all the physical behavior, all the dependence on the lag time factorizes. And all the influence of this aging stuff becomes a prefactor. So unless you care for the exact coefficient, you can neglect the aging in the measurement. And that's a beautiful result. When Johannes, my PhD student, got that, this was a beautiful one. And PRL thought the same. And what you, for instance, then can calculate is the probability that during your measurement time, capital T, your particle makes at least one step. It's decaying like this power law. So what in essence you will find is that your, when you look at the number of jumps during a certain time window, you have a splitting of the population into a discrete part that does not move at all, and then another part they have different mobility. And that's what you also see in these simulations. Here this would be a continuous time random walk. No aging, you see the big scatter we had before, the linearity in delta, and you see essentially all the simulated trajectories here. When you age it, we have the same number of simulated trajectories, but a lot of them are somewhere down there, they don't move. And if you then calculate the real average over all the trajectories, this would be the mobility, or the mean. Whereas when you look at the data that are non-zero, maybe your measurement doesn't see them, right? You would guess a completely different amplitude for this. So when we have processes like these continuous time random walks, there are others that can be even more extreme. It very much highlights the importance of knowing what you fit to. You need to know these models to extract proper quantities. For instance, here you would completely overestimate the mobility of the particles. That's all I see. So here is another example of aging. This is really going back to these amorphous semiconductors. One of my colleagues can actually measure these guys. These are now polymeric semiconductors, and what they can do is they measure the electrical current in a slab of these semiconductors. Initially, they put these charge carriers in there by a light flash, but then they let them move freely for an aging period, and only afterwards they switch on a driving field to a counter electrode, and then they measure the characteristic of the current. And in this log-log plot, the black line up here, this is without aging, that's the old Sherman-Troll result, where you cross over from one power law to the other, and the sum of the two power laws is exactly -2. But when you age the system more and more, you actually develop a plateau, and you barely see it here. You get an intermediate, the calculations show that there's an intermediate scaling range, and then you get to a terminal scaling. And from simulations, they actually, I don't have the image here, they extrapolated these plateaus, and then when we take the values of the plateaus and we match them with our theory, with the measurement, with the aging time, you actually see that there's a beautiful. Good. And the last thing about aging I want to tell you is something that thrilled me recently when I saw this. So this is a combination of very large-scale simulations results in the group of Jeremy Smith at Oak Ridge, and experiments that were done in the, I think they were published in 2003 by the group of Sunny Shi. So what do? They have a single protein. No, they have water, but nothing else of the cell. There's no crowding here, there's nothing. It's a single protein embedded in water, and they actually look at the relative distance as a function of time between two monomers in the protein. So for any major relative motion of these two guys, the rest of your protein chain in between, all these individual monomers, they have to reconfigure. So there's a very, very complex hyperdimensional configuration landscape. And when they measure the motion of this guy, I don't want to go into all the details, they see anomalous diffusion, and mainly they see aging. For instance, here in the correlation function, you see it for different measurement times. You see that the timescales encoded in the correlation function actually are measurement time dependent, so it's an aging process. They show that over 13 orders of time, combined with the experiments and the simulations, they see this non-stationarity, they see aging. And we're talking about a single molecule. So I was really fascinated by that. And they interpret it when they look at the configuration landscape. This is only a small fraction of the configuration landscape, but then they identify specific configurations in which the protein is almost trapped. It's very difficult to get out from these specific states to get to other configurations, and they attach actually power law waiting times to these states. So even on a single molecular level, we have these types of motion. And if we look here, we have time scales. This is 10 to the minus 12 seconds. Here we have 16 or 15. So we have actually 1,000 seconds, which is quite remarkable.
Can you go from the trajectories there to figure out the landscape structure?
I mean, they got the by landscape you mean
That sort of energy surface there, I guess, or the folding energy surface.
maybe, because to visualize all these, you always have to define a reaction coordinate and that stuff, and I think that's the tricky part. They didn't do it in the paper. I didn't ask for it as a referee, I have to say, because I don't like these landscapes too much. Essentially, you have all the coordinates from the simulations. So essentially, you should be able to look exactly where this motion takes place and then assign at least the local landscape. Yes. So I have all kinds of other stuff that I will just flip over. Let me show you one example of super diffusion. This is now in a very complex cell. Do I have 2-3 more minutes? Do you know?
3 more minutes. Let's say 4.
Good, fair enough. So very, very quickly, I was talking mainly about membranes. When you look into the liquid of the cells, this is actually what it looks like. This is a computer simulation from Adrian Elcock's lab. These are the 50 most abundant proteins in a bacteria cell. And we're talking about chunky, big molecules, and they occupy around 35% of the volume. This is big. Remember, it's 3 dimensions. Cubic percolation, the threshold is 31%. So 35% is a big number in that sense. And then you get all these anomalous diffusion in those cells, but a friend of mine, Christine Selhuber, in Kiel, she's actually looking at even more complicated cells where you have all these vacuoles and granules and whatever. We call that super crowded. And what you see in these cells is that you have anomalous diffusion of passive particles, but in addition, there are whole swirls of the whole inside of the cell. When you do single particle tracking, in that case, you actually see super diffusion. So this is normal diffusion, this is ballistic, so you see behaviors in between. And this is, of course, then a question when you want to think about modeling of cellular processes. Does this speed up mixing in the cell? Good. Let me jump across and let me use my last and final minutes to highlight a new section. You're all into maths here, so I think it's appropriate. So at the Journal of Physics A, we started a new section that's called Biological Modeling. It's a British journal, so we have 2 L's here. It's actually a section that's catering for all those papers that are considered too mathematical for the other journals. I saw that Journal of Theoretical Biology is not doing that well, actually, I was surprised. All are not considered general enough for any of the other journals. So if you have any paper, or if you have an idea for a review or a special issue, contact us. So we're trying to nurture this new section. We have the first papers coming up there, and I'm very happy that this step was made. So we have all this anomalous diffusion in the lipid bilayers, and I showed you that when we increase the disorder from the pure lipid case, including proteins or cholesterols, going over to the real natural case, we expand the anomalous diffusion to macroscopic scales, and this would be a new simulation that's being prepared for a really crowded membrane, where we have different proteins embedded. We get non-Gaussian diffusion when we crowd the bilayers, when we have these many proteins in there, and we get even this continuous time random walk style motion and aging and non-ergodicity in the real biological cells. What I didn't show you, when, for instance, we look at this, the effect of the viscoelasticity of the environment, and we look at the molecular motor pulling a big chunky cargo, you also see anomalous transport, so that your mean position of the motor and the cargo actually becomes a power law, so you also have effects there. And what I didn't talk about, there's all kinds of other processes where you see violation of ergodicity and that are relevant for certain systems. For instance, I mentioned time-dependent diffusivities. That would be a good model for granular gases when people more or less fuzz about with breakfast cereals, right, and vibrating tables. There you have collisions between the particles and they lose kinetic energy, and so this kind of effective temperature is going down. It's very similar, it's very easy to confuse it with a continuous time random walk, as a matter of fact, when you look at the data. Thank you.
Okay, thank you, Ralf. Further questions? Peter.
So I wonder whether you consider this simple model sort of relevant to the aging you're talking about. So if you take critical branching process, right, then on average, the ensemble average is the population's 1. Of course, it happens, there's a probability of 1, everything goes extinct, but condition on going extinct, the size is growing. So one of your last slides, we sort of had like the distribution of things that weren't, you know, trapped or whatever. You would have calculated the wrong mean square displacement based on that. So other than the fact that this doesn't have a power law in it, is this the same basic concept of aging as what you're describing?
Maybe. I need to think about it.
Okay.
When you talk about extinction, I could even imagine something when you think about finite time singularities and you put noise around them and whatever, then you could probably create something like that as well. But that works. I need to think about it.
Yeah. In polymers like chromosomes, or I mean, also interested in your protein, well, not your, but the protein simulations that you talked about. What do you expect there? I mean, the theory is also that it's Gaussian, but I mean, but
in chromosome in
yes.
chromosome territories, whatever it's.
not territories, but like if you, yeah.
So what measurements show you? A friend of mine, Yuval Gavini, does measurements on these things at Birmingham University, and what they say is that when they look at the motion of the telomeres, the end parts, and they can also map these loci somewhere inside the chromosome, they more or less see this fractional Brownian motion. They didn't check whether it's really Gaussian. So what they check is they see this anomalous diffusion, they see that they have these negative correlations, but we had them for this protein crowded case as well, just that we were able to look at the PDF. So they don't have enough data to do that. So whether it's really Gaussian, I don't know. But it looks like a godic, it's anomalous. I can show you some
Yeah,
pictures of this.
All right, let's stop here. And before ending this session, I just would like to say that now, for those of you who are interested in these European grounds, we'll have a meeting in the Newton Institute. So let's thank Ralf again. Thank you.